In the second half of Chapter 6, we talk about the reactions that are part of photosynthesis. There are two reactions that occur in photosynthesis: the light reactions and the so-called Calvin cycle reactions, also referred to as the light-independent reactions. If we look at the screen, on the left-hand side of the screen, we see the light reactions. On the right-hand side of the screen, we see the Calvin cycle reactions. Now, both of these have to occur for photosynthesis to take place, but they play different roles in this process. We begin with the light reactions. The light reactions produce two products that are essential for photosynthesis: NADPH and ATP. These products play different roles in the process of photosynthesis. NADPH is an electron carrier. ATP is, of course, energy, as you already know. That energy is going to be incorporated into the sugars, and the electrons carried by NADPH are also going to be incorporated into the sugars. The Calvin cycle reactions are where the sugars are actually produced. And so as we see in our notes here, CO2 is required, and the products are carbohydrates, and the products of the Calvin cycle reactions. So carbon dioxide goes in, we add ATP, we add NADPH, and then from the Calvin cycle, we get ADP and NADP+. NADP plus is an empty electron carrier, and as you know, ADP, adenosine diphosphate, is ATP that's lost that third phosphate and therefore transferred its energy. It is a low energy molecule. So, to review, the light reactions require solar energy, and they produce two things: NADPH and ATP. They occur in the thylakoid membrane of the chloroplast. The Calvin cycle reactions occur in the stroma, outside, as is shown in this diagram, outside the thylakoids. They require NADPH from the light reactions and ATP, and the Calvin cycle reactions produce carbohydrates, in other words, sugars. We spend just a moment talking about the Calvin cycle reactions here. Honestly, we covered almost all these topics on the previous slide, at least the top 3 points. The Calvin cycle reactions are powered by ATP and NADPH, as we mentioned previously. ATP provides energy, NADPH provides electrons. The Calvin cycle reactions occur in the stroma, and the end product of the Calvin cycle reactions are glucose. There are 3 steps that are involved in the Calvin cycle reactions. One is CO2 fixation, one is CO2 reduction, and one is regeneration of the first substrate, which is RUBP, or ribulose 1,5-biphosphate. Now, we need to understand what this word means: fixation. In biology, this means you take something in a gaseous form and you turn it into a usable form. In the case of CO2, we're taking carbon dioxide from the atmosphere and turning it into a sugar. You, for instance, can eat a sugar and it becomes part of your body and you use that for energy. You cannot consume CO2. You inhale it and exhale it, but it does not interact with your body. CO2 reduction means this: the addition of electrons. CO2 reduction is the building of sugars by the addition of electrons to carbon dioxide. And then lastly, the regeneration of that substrate must occur for the Calvin cycle, implying that it's a cycle that happens over and over again, to occur. So here we see a brief picture of the Calvin cycle. It's not our task to learn all these little parts at this point, but I'm going to point out a couple little parts that we do need to know. So the very first thing we notice is this: CO2 is going in here. What is this called? We remind ourselves it's called CO2 fixation, the addition of carbon dioxide from the atmosphere into a solid form, in other words, into a form of a carbohydrate. Here we're adding ATP, here we're adding electrons, here we're adding ATP, and then eventually we regenerate that original substrate. And so RUBP, ribulose 1,5-biphosphate, is the original substrate. RUBP has 1, 2, 3, 4, 5 carbons, those grayish spheres, and we're going to add 1 carbon dioxide to that. I know we had 3 total, but 1 to it, and we get this 1, 2, 3, 4, 5, 6 carbon intermediate that splits into 2 3-carbon intermediates. So what we notice is this, that we have to add 3 carbon dioxides, and this cycle has to turn 1, 2, 3 times before we remove 1 carbohydrate. The carbohydrate removed is referred to as G3P, glyceraldehyde 3-phosphate. And so that's the basic idea of the Calvin cycle. We are adding carbon dioxide, in other words, we're fixing carbon dioxide, then we are reducing what we added. So here we're adding electrons. The addition of electrons means reduction. Or reduction is the addition of electrons. And so as we add these electrons, we're reducing CO2, and eventually, after we turn 3 times, we create a carbohydrate. So we put in ATP, we put in NADPH, and we get out a carbohydrate. What we notice is that we have 3 types of photosynthesis that we want to mention, and the 3 types of photosynthesis are perform better or worse in different types of environments depending on the amount of rainfall that's received. Let's take a look. The first type of photosynthesis is C3 photosynthesis. In C3 photosynthesis, then, interestingly, carbon dioxide enters, this is the Calvin cycle, of course, and becomes part of this cycle, and then the carbohydrate leaves, and nothing else is involved here. And so there is an enzyme called rubisco, one enzyme called rubisco, and rubisco adds CO2 to the Calvin cycle. Its responsibility is to fix CO2, to add it to the Calvin cycle. This is fine, but what we notice is rubisco is not very efficient at grabbing CO2 and adding it to the Calvin cycle. And so we have to have very high concentrations of CO2 for it to work. So C3 plants do extremely well as long as they can leave their stomata open. And you can leave your stomata open, but the problem is that you lose water. So as long as there's plenty of rainfall, then C3 plants do extremely well. However, if you have a drought, then C3 plants start to suffer because they need to close their stomata. When they close their stomata, they can no longer carry out photosynthesis. So they're in trouble. That brings us to C4 plants. In the case of C4 plants, we have an extra step in the process of photosynthesis. C4 plants possess an enzyme called PEP carboxylase. PEP carboxylase. PEP carboxylase grabs CO2 and fixes it, creating a 4-carbon molecule. Another enzyme then removes that carbon from C4 and feeds it into the Calvin cycle via the enzyme rubisco, just like we saw happen in the case of C3 plants. The difference here is that this enzyme, PEP carboxylase, that grabs CO2 and adds it to the C4 molecule, is very, very efficient. So it can continue working down to very, very low concentrations. So C4 plants have no trouble closing their stomata and continuing to carry out photosynthesis because they could still grab those CO2 molecules even at the very low CO2 concentrations that are found in their cells after they have closed their stomata for an extended period of time. On the page, we see a corn plant. Corn plants are C4 plants. They do very well in very warm, fairly dry conditions. On this page, we see the contrast between a C3 and a C4 plant. This is crabgrass that I'm pointing to here on the right-hand side of the diagram. On the left-hand side, we have fescue, which is a C3 plant. And so this picture is taken in August in a very hot, dry time, and the yard was mowed the yard was mowed, and then these plants grew. And so what you notice is the C4 plant, the crabgrass shown on the right, is far outpacing the C3 plant, fescue, in terms of the growth rate. But this only happens during the warm months. If you back up to April, then both of these plants would have been growing at about the same rate, or the C3 plant would have been growing a little bit faster. So interestingly, C3 plants have an advantage in cool, moist conditions. C4 plants have an advantage in very hot, dry conditions. Lastly, our final form of photosynthesis is CAM. which stands for crassulacean acid metabolism. It's named after the plant family in which this type of photosynthesis was first discovered, but now it's known that this type of photosynthesis occurs in many different families of plants. What we see is that this is almost identical to C4 photosynthesis. However, in this case, the plants only open their stomata at night. At night, the humidity levels are higher, and they lose less water when they open their stomata at that time. The downside is they cannot carry out the light reactions at night, and so they have to wait for the sun to come up to carry the light reactions. This means they store the carbon dioxide in this C4 molecule. When the sun comes up, they start to run the Calvin cycle when they have the NADPH and ATP produced by the light reactions. So this is good and bad. It's good because it allows them to live in very dry environments. They only open their stomata at night. They lose very little water. It's bad because they cannot store too much. There's a limited volume of CO2 that they can store in this C4 molecule. This means that they are limited in their overall growth rate because of the slow rate of photosynthesis. But that's okay because they live in such dry environments that it's difficult for other plants to survive there, either C4 or C3 plants. A pineapple and is a good example of a CAM plant. And many desert plants are also good examples of CAM plants. So CAM plants are adapted to the driest and the hottest conditions, even more so than C4 and certainly far more than C3.